Okay, so um good day everyone. So welcome to our developmental psychology class. So today we're gonna talk about the lifespan development um in the prenatal development. So we have here module 3, prenatal development. So let's start. So um in this module, we are about to explore the journey of preparing for birth and the development process of the from conception to delivery. So development process from um, conception to birth. Okay, so our outcome should be or should explain the role of genetics in prenatal development. So next we have to explain the main stages of prenatal development and of course describe the approaches to childbirth and the labor and delivery process. So these are our learning outcomes. Okay. For our first topic, we have the biological foundations of human development. So here are the like minor outcomes based from this prenatal development topic. We have explained the role of genetics in prenatal development, explain the evolutionary psychology perspective of lifespan development, describe genetic components of conception, describe genes and their importance in genetic inheritance, describe chromosomal abnormalities, explain the value of prenatal testing, describe the interaction between genetics and the environment, and comparing monozygotic and dizygotic twins. So, going into our discussion, we have here the term evolutionary psychology. So, when we say evolutionary psychology, it focuses on how universal patterns of behavior and cognitive processes have evolved over time. So, this is the belief that we, as a psychology major, believes that there is a universal pattern of behavior and cognitive processes, no, that is evolving over time. So evolutionary psychologists study a variety of psychological phenomena that may have evolved as adaptation. So evolutionary psychologists believe that this cognitive or behavioral patterns that changes are a indicator that we are adapting, no, to the present, for example, present need or scenario for us to survive. So therefore we have here the term the theory of natural selection. This is according to Charles Darwin, no, the father of evolutionary theory. So it refers to the survival and reproduction of organisms better suited for their environment over those poorly suited. So this theory, which is natural selection, says that um, whoever or whichever organism that is has the capacity to better reproduce, no, or survive because of its capabilities, will have a better, you know, adaptation or better a longer lifespan, should I say, if they can reproduce and they have the ability to survive. Okay, so evolutionary psychology enhances our understanding of development through how we choose mates and achieve reproductive success. So evolutionary psychology um, lets us understand, no, on how it is important for us to choose mates because as we know, um, in order for us to survive, we need to reproduce. Okay, so maybe number dictates survival for this one. So we must achieve reproduction, and we must choose um, you know um, a very good mate or partner relationship as well. So next we head on to the heredity and chromosomes. So there are two types of gametes involved in reproduction. We have the male gametes or sperm and the female gametes or ova. So um, gametes or sex cells. No, they're also called sex cells. We have sperm for male and ova for females. Yeah. So male gametes are produced in the testes through spermatogenesis beginning at about 12 years of age. So um, male gametes or sperm are produced with the process of spermatogenesis. So this is the process of generating or producing sperm in the process of spermatogenesis. So this starts about 12 years of age of a male. No. So next we have female gametes are stored in the ovaries and are present at birth, though immature with only about 400 becoming mature eggs. So however, in females, it is it has always been in their ovaries, no, upon birth. So however, these eggs or ova are not yet matured, no, and it only has 400 uh, that are becoming mature eggs. So once a ripened egg is released from the ovary, it moves into the fallopian tube and reaches the uterus in the 3 to 4 days. So a ripened or a mature um, ova will be released in the um, ovary, and then it will travel along the fallopian tube until it reaches the uterus within 3 to 4 days. So and then after that, once a single sperm unites with the nucleus of the egg, a new cell with the combined genetic information from both parents is a zygote. So upon the contact or upon the fertilization of the sperm and egg cell from the fallopian tube, it will form a what do we call a zygote. So it is the combination of egg cell from females and sperm cell from the males. So it will form a new cell that is called zygote. So we have here the term chromosome. So chromosomes are long strings of genetic material known as deoxyribonucleic acid or DNA. So ito yung parang mga hagdan-hagdan na genetic makeup natin, no, as a human, or even other organisms have specific structure of their DNA. So here's an example of DNA. We have the base pairs, as we can see here in the image. No, it's like a ladder, spiral ladder. Okay, and it has steps. Okay, so we have base pairs, the combination of adenine and thymine, and combination of guanine and cytosine. So at the edge of each steps, we have a sugar phosphate backbone. As you can see, the blue spiral flat thing around the our DNA. So DNA is a helix-shaped molecule made up of nucleotide base pairs, adenine, guanine, cytosine, and thymine. So we have AGCT. So adenine guanine, cytosine, and thiamine. Adenine guanine, cytosine, and thiamine. So each chromosome has a sequence of DNA makeup genes that control a number of visible characteristics. So each gene has a multiple variations of alleles, specific version of genes. So alleles are specific versions of genes wherein these can be manifested or unseen, no? So later on we will dive into those. So in meiosis, a segment of chromosomes from each parent form pairs, and genetic segments are exchanged as determined by change. So meiosis is a process of pairing of genetic segments from the parents. Okay, so meiosis, process of pairing of the genetic segments from the parents. Okay, so each parent contributes half of genetic information carried by the offspring, with the resulting characteristic determined by interaction of parental genetic material. So according to this passage, we have a 50 to 50% of the genetic information comes from our mother and father. Okay, so we have here the genotype. Genotype is the genetic makeup of the individual, while the phenotype is refers to the person's inherited physical appearance and characteristics. So what's the difference between genotype and phenotype? When we say genotype, it talks about the genetic makeup of the individual, while phenotype refers to the inherited physical appearance. So basically, phenotype we can observe this. However, genotype we cannot observe the genetic makeup unless we use certain apparatus or instrument for us to determine those genetic makeup. Okay. So next, we have 22 of chromosomes from each parent are similar in length to a corresponding chromosome from the other parent, with the remaining chromosomes looks like an A, X, or Y. So we have 23 pairs of chromosomes to each parent. So each offspring has 23 pairs of chromosomes. So 22 from those are similar in length to corresponding chromosome from the other parent, with the remaining chromosome. So 22 are similar. And then the 23rd pair determines whether the offspring is a male or a female. So it can be X or Y. So we have here the letters XY for male and XX for female. So take note of that, huh? So we have XY male and XX female. Okay, so next we have genetic inheritance of traits for humans is based upon Gregor Mendel's model of inheritance. So our genetic inheritance of traits for humans, no, is based from Gregor Mendel's model of inheritance. So we also have here autosomes. So any chromosome other than the sex chromosome have alleles, and their associated traits are autosomal dominant or autosomal recessive. So autosomes are other cells, that are not sex chromosomes. So when we say autosomes, they are not sex chromosomes. So they are associated with dominant or uh, autosomal dominant or autosomal recessive. Okay, so autosomes, non-sex chromosomes. So we have here a sample of a Punnett square. So this would determine the characteristics or phenotype of an offspring. Okay, so we have here the capital N, and then it will produce double capital N. So you will just multiply it or combine those phenotype in order for you to determine the percentage of having a um, dominant trait or recessive trait Yeah, and those things. So we have here the genetic variation and inheritance. So we have dominant genes. So dominant genes are expressed while recessive genes are only expressed in the absence of a dominant gene. So when we say dominant gene, these are the manifestation of the traits from the parents to the offspring. So dominant genes are directly observed. However, recessive gene are not our physical appearance or characteristics that are not expressed unless it is paired to another recessive gene. No, so recessive plus recessive, it will be expressed as a dominant gene. Okay, remember that. So if a person has only one recessive gene for disease, they would be a carrier. So example is cystic fibrosis. No, someone is homozygous for an allele when they have two copies of the same allele, and heterozygous when they have a combination of alleles. So we have here different kinds of alleles. We have the homozygous and heterozygous. When we say homo, means same, no, so homo means same allele, and hetero means many or combinations of allele. So homo, same allele; heterozygous, a combination of alleles. So next we have harmful genes like cystic fibrosis comes from a gene mutation or sudden permanent change. So harmful genes, cystic fibrosis. So cystic fibrosis is an example of gene mutation. No, it can be harmful. Okay. Okay, next we will talk about the chromosomal abnormalities and genetic testing. So here in the figure we can see the three major single chromosomes: mutation, deletion, duplication, and inversion. So we have the number one is deletion. So those chromosomes here are not present to the uh, second chromosome. In number two, we have duplication. So instead of one chromosome, it became like two chromosome, no? So it duplicates. And inversion is the retention, inversion or retention. So it just moved from one chromosome to another chromosome. So it just moved. Inversion or the what do you call this, yeah, inversion or the movement of chromosome to the same. So chromosomal abnormalities occur when chromosome structure is altered and includes, okay, so um, uh, this abnormalities happens when the chromosomal structure is altered. So these are examples of alteration of the chromosomal structure. So we have deletion, portion is missing or deleted. Next is duplication, portion is duplicated or multiplied. Instead of one, you have two. Next is translocation, so portion is transferred to another. So uh, the difference between translocation is and inversion, no. Translocation, the chromosome is transferred to another chromosome, and from there it is gone, no, the portion is transferred to another. However, in inversion, a portion has broken off and retouched upside down. So in inversion, it duplicates, I know, it um inversed, no, meaning to say it is rotated, no, portion is broken off and reattached upside down. So instead of, you know, retaining its um position, no, it transverse itself upside down. So next we have insertion. Insertion, portion has been deleted and inserted into another chromosome. So insertion is um, you've inserted the chromosome inserted from another. Okay, so We have here the rings portion is broken off and formed a circle. So it broken down and then formed a circle. Next, we have isochromosome formed by by the mirror image copy of chromosome segment. Okay, so one of the most common chromosomal abnormalities is pair 21 or the Down syndrome. So Down syndrome or the chromosomal abnormalities on the pair 21 of chromosome, also called as trisomy 21. So that is a Down syndrome. We have here a chromosomal abnormality on 23, results in a sex-linked chromosomal abnormality. So abnormality in number 23. Okay. Turner's syndrome occurs 1 out of 2,500 live female births when an ovum lacking chromosome is fertilized by a sperm with an X chromosome, resulting in a zygote with an XO composition. So Turner syndrome is missing X with the female um, gene. No, we have here XO composition instead of XX, no, sa females. Next is Klinefelter syndrome. XXY occurs when 1 out of 5 male births. So extra X chromosome is fertilized by the Y sperm. So in males, we must only have XY chromosomes. However, if the Y sperm is or have fertilized another X chromosome, then it would be Klinefelter syndrome or XXY um, chromosome. Okay, you have another extra X chromosome. So next we go on to the prenatal testing. When we say prenatal testing, here are the process on which the screening focuses on finding problems among a large population with affordable and non-invasive methods, and commonly includes ultrasound, blood tests, and blood pressure readings. So prenatal screening, so this would be a process of determining whether you are whether a female is pregnant, no, by the use of ultrasound, blood tests, and blood pressure readings. I will not agree that these are non-invasive methods because sometimes, no, these processes such as ultrasounds and blood tests may cause miscarriage or um, other things that will be negative or that will produce a negative effect on the uh, development of the neonate. Okay, so prenatal diagnosis focuses on pursuing additional detailed information on a particular problem and has been found can be invasive. So diagnosis screening can detect problems such as neural tube defects, chromosomal abnormalities, and gene mutation. Prenatal diagnosis is intended to enable timely medical or surgical treatment of condition, to give parents a chance to abort fetus, and to give parents a chance to better prepare for a baby. So, basically, pre-testing is done on the actual, no, screening if a female is having a pregnancy, and then after that they would determine, no, whether the infant or the neonate has abnormalities and such defects, no, in order for the parents to prepare, or if ever to abort no a fetus and to give them a better preparation for the baby's special needs. So next we have here behavioral genetics. The nature and nurture debate questions the role of heredity versus the environment in our personality. So nature versus nurture. This has been a long debate in the field of psychology or behavioral genetics. Um, whether we are nature or we are made from the social interactions or environment for nurture. No, so nature says that we are predetermined. No, all of our characteristics, behaviors, cognitive um ideas are based from our genetics. No, we have inherited this from our parents, so that's nature. We have been predetermined. However, in the nurture part, says that we are a product of our environment, that all of our thoughts, feelings, emotions, actions are shaped and influenced by the world around us. So most scholars agree that there is a constant interplay between nature and nurture. While genetic predisposition are important, other factors are in a part of the effect of epigenetic above genome and changes. So epigenetic describes psychological development as a result of an ongoing bidirectional interchangeable between heredity and environment. So we have here the term epigenetic principle, wherein um, the nature and nurture debate no becomes one. So it says that our development is a result of an interaction or interplay between our genes or heredity and our environment. No, behavioral genetics is the science of how genes and environment works together to influence behavior and can be studied in adoption studies and twin studies. So behavioral genetics. So here in adoption studies, it talks about um, people who are adopted, or sometimes they have uh, I here in adoption studies and twin studies, it studies about an offspring or twins that are maybe adopted or living in the same house. So for those adopted twins, they've studied that they may have same characteristics, behaviors, emotions, and perceptions, even though they are not living together. No, so that dictates, or that um, gives an idea that development is based on the nature. However, we have other um, twins that are being separated from each other or even living together, but they have different no they have different styles, different ideas, perception, behavior. Even though they are twins, they are living on the same house, they still have differences. So that's a nurture debate. So sometimes we, even though we grew up in the same house, we still have different experiences. No, we still have different scenarios that we experience throughout the day or throughout our lifespan. So therefore, it says that we are nurtured, or our environment shapes us. No, so we still have behavioral genetics, practice questions. Okay, so let's finish this muna. So We have here the monozygotic and dizygotic. So monozygotic is a single zygote or fertilized egg splits apart, and then dizygotics naman are fraternal twins, same genetic material, same mother and father. So monozygotic has a single zygote or fertilized egg, and then it splits apart. So meaning to say mono, just a single egg, but creates two separate identical offspring. However, in dizygotic twins, there are two eggs and then two sperm, no, fraternal twins that has been fertilized, no, sa dizygotics we have two. So in the uterus, 60 to 70% of monozygotic twins share the same placenta, a temporary organ collecting figure. So placenta is a temporary nutrient supporting organ ba siya? I don't know, but it is a part of the um, development wherein it becomes a temporary uh, source of nutrients of the fetus, no, it and it is attached to the uterine wall but separate amniotic sacs, a thin but top transparent pair of membrane that holds the developing embryo fetus until shortly before birth. Okay, so we have here conjoined twins. Bodies are joined during pregnancy, and vanishing twins, as many as 1 in 8 pregnancies start out as multiple but only single fetus is brought to full term. So, vanishing twins, mayroon daw siyang twin, but then only a single fetus is brought to full term. So, twin studies, just like what I've said earlier, a behavior genetic research method that involves comparison of the similarity of monozygotic and fraternal twins. So our practice question, we have Shelby is taking with her doctor about prenatal screen. She wants to know what will be covered in the screening, which of the following is not a conditional prenatal screening tests routinely screen for. So genetic disease, birth defects, cancer, chromosomal abnormalities. You have 3 seconds. So the answer is C, cancer. No. So prenatal screening is not made for detecting cancer. So it is made for genetic disease, uh, screening and birth defects, chromosomal abnormalities, but not cancer. Question 2. Which of the following is not considered a recessive trait or disease? So recessive trait or disease, we have red hair, nearsightedness, Huntington's disease, male pattern baldness. So we have 3 seconds. So it is male pattern baldness. So red hair is a dominant trait. Nearsightedness is a disease. Huntington is all of course a disease, but male pattern baldness is not considered a disease, and it is recessive because it is not directly observed during early childhood, but then it develops, or um, it happens during your adulthood stage. So it is a recessive trait. It's still there, but you cannot directly observe it in an instant. Okay, so that's it for our heredity and genetics and the topic prenatal development, and our next topic would be prenatal development.